Okay, in the last class we were discussing about oogenesis. In oogenesis we studied two different processes. Number one is how the follicles are getting developed. So that is from primary follicles. Before primary follicle we had primordial follicles and then primary follicle and then secondary, tertiary and graphene follicle. Tertiary follicle will get matured into graphene follicles. Okay. Simultaneously, we also studied about the structure of the ovum and how the ovum is developed. So we started with the oogonia, otherwise it is called as egg mother cell. So this oogonia or the egg mother cell will differentiate, will undergo a division called mitotic division to form primary oocyte. And everything is done before the birth. A female is born with primary oocytes. It is around 1 to 2 millions. Now it is arrested till the time of puberty. By the time the primary follicle will develop into secondary, tertiary and graphene follicles. Now the primary oocyte will attain or undergo a reduction division called meiotic 1 or meiosis 1 and it will produce the secondary oocyte. It is not only producing secondary oocyte, primary oocyte will undergo meiosis 1 to produce one large secondary oocyte which is haploid. At the same time, it is also produces one small haploid first polar bodies. So first polar bodies are formed from the primary oocyte during the development of secondary oocyte. Then it is arrested till the female is undergoing fertility. When a girl attains fertility, this secondary oocyte further undergo second meiotic division to form another two cells, that is one large haploid ovum and the second one is the second polar bodies. These two polar bodies are disintegrated. So at the end of oogenesis, we had one haploid ovum finally, and the polar bodies disintegrate. So end of this oogenesis, we had two polar bodies. Okay. Then we studied about the structure of ovum. Structure of ovum, when we talked about our human egg is a non-cleidoid. It do not have any shell at all. And it is alecithal or microlecithal. Why? Because they have no yolk or little. The placenta will be supplying the nutrition for the developing fetus. Then the egg will have a center. It is having in one corner. It is having germinal vesicles in which a nucleus will be there, which is undergoing enlargement, and it is filled with ooplasm. And the egg cell itself will make a membrane called Vitellian membrane or otherwise called as egg membrane. Clear? Now this the nature of this Vitellian membrane is inner membrane and it is very thin membrane. Clear? And one more, the membrane which is made outside on this Vitellian membrane, it is very thick, that is called zona pellucida. It is a primary egg membrane. We will talk about it later. Then it is also forming a radial shaped cell which is another thick, that is the corona radiata. Okay, in between the Vitellian membrane as well as the zona pellucida, we find a space called perivitellian space and filled with a fluid called perivitellian fluid. Okay, right. So that is what we studied in the last class. Clear? Today we will be concentrating on the spermatogenesis process. Yesterday we talked oogenesis. And today we are going to talk about spermatogenesis, that is the formation of sperm cells. Formation of sperm is called as spermatogenesis. Before we could study the spermatogenesis, you should understand certain terms in this. That is, one is called this spermatogenesis. The first one. Second, we will talk about spermiogenesis. Okay. And the third one is the spermatid. And the last one is the spermatozoa. So these words we will come across. Everywhere you will find sperm, sperm, sperm. But still there is a confusion. That is the reason you have, particularly when you study this question, you have to write and then you have to study. Then only you will not make any mistake of it. Okay. Now we will concentrate this spermatogenesis now. Spermatogenesis here, it is the production of formation of sperm cells, but it is always immature process. The it is the immature process which will get into maturation process. So the immature process is the spermatogenesis, and the maturation process is called spermiogenesis. Okay, so this process is called the maturation process. Understand what is spermatogenesis? Immature sperm cells are produced. What is spermiogenesis? So in spermatogenesis, in production of the formation of the sperm cells, it takes place in two processes. One is the spermatogenesis and then it becomes spermiogenesis. The last class we discussed about the female are born with very beautiful. Female are born with primary oocytes. Okay, then it is being arrested. After puberty only it starts. Male are born with a cell called spermatogonia. Okay, spermatogonia is a sperm mother cell. It's a primordial cell. Clear? Female born with primary oocyte, male born with spermatogonia is a sperm mother cells. Okay. Now, in human male, this spermatogenesis starts only at the time of puberty. No before, no after, nothing. So at the time of puberty only, the spermatogenesis process takes place, formation of sperm cells. And that too 10 to 16 years. Clear? Now, this spermatogenesis will takes place in the puberty. Why? Because it requires two hormones. First one, it is in the, we studied the hormones a lot. In hypothalamus, we add a releasing factor. You remember? Hypothalamus will always have releasing factor. So this has an hormone called gonadotrophic releasing hormone. We studied what is the difference between gonadotrophic hormone and gonadal hormones. Anything that is there in the pituitary are called gonadotrophic hormone. Anything that is in the reproductive organ is called gonadal hormones. Clear? Right. So now this spermatogenesis, why we say it is beginning in the puberty, but still it is not very clear whether it is starting in the puberty or not, before or not, but still we believe because for spermatogenesis you need the function of the hormones. That is the reason. So now this hypothalamus will give out a hormone called gonadotrophic releasing hormone. Okay, and this will enter into the gland called the pituitary gland for producing FSH hormone as well as LH hormone. Okay, follicle stimulating hormone and luteinizing hormone. Now, this follicle stimulate, now the role of the luteinizing hormone is, it will act on the cell which is there in the testis called Leydig cells or interstitial cells, and it will produce an hormone called testosterone hormones. So, LH will involve in producing an hormone called testosterone hormones. Whenever you have this hormone, spermatogonia is produced. Okay, so now what is the role of the testosterone hormone? Will always involve in the process called spermatogenesis or spermatogonia is formed. So spermatogonia is formed. So the process is called spermatogenesis. So testosterone hormone will always involve in the spermatogenesis, that is the production of the spermatogonia, that is sperm mother cells. Okay, so here we study formation of sperm. That means it is a process which is evolving in it. So here it is the spermatogonia. Is that clear? So now this gonadotrophic releasing hormone should go to the pituitary and stimulate this hormone called LH. Now the LH will act on a cell which is there inside the testis called Leydig cells for producing an hormone called testosterone, and the testosterone will involve in the spermatogenesis. Immature sperm cells are produced. Is that clear? Right. Now, FSH hormone will always involve in acting or stimulate a cell. One more cell is there inside the testis, that is called a Sertolic cells. Okay, this is the cell which is there inside the testis. Okay, so now this FSH hormone will involve or stimulate the Sertolic cells to produce an hormone. Sertolic cell will produce. Actually, it is an endocrine gland. You remember, it is not there in the book, but still, this can produce an hormone called inhibitin. Okay, it is an hormone which is produced by Sertolic cells called inhibitin. That is, whenever the testosterone level is increased, that time this FSH hormone will act on this Sertolic cells. It will produce an hormone called inhibin or inhibitin. In some book you will find inhibin. Some book you will find inhibitin, but this hormone will inhibit the production of testosterone. Is that clear? Then it is also having a protein called androgen binding protein. Androgen is a hormone binding proteins. Okay, so otherwise it is called ABP. ABP, androgen binding protein. So this androgen binding protein is produced from a cell called Sertolic cells. This androgen binding protein will always maintain the concentration of testosterone. Understand? If the concentration is maintained, the spermatogonia is produced. If the concentration goes high, it will stop by a factor called or a hormone called inhibitin. This much is clear. So, if this thing should happen, then only the spermatogenesis will take place. That is the reason we understand the spermatogenesis takes place at the time of puberty, not before. Got it? So that is what we are studying here. Now we are concentrating on what is spermatogenesis. In spermatogenesis, we are discussing about the formation of sperm cells, how the sperm cells are produced. The spermatogenesis are production of the immature sperm cells. Okay, right. So now these immature sperm cells are produced at the time of puberty. Why? Because the hypothalamus will give out the releasing factor called gonadotropic releasing factor. It will enter into the pituitary gland and stimulate the two hormones. One is called FSH and LH. LH hormone act on the Leydig cells. One more name is there, interstitial cells. For this, it is Leydig cells also called as interstitial cell. That means in between. Interstitial means in between. There are two tubes are there, means in between the tube. That is called interstitial. Okay, or Leydig cells. So it will act on that and produce a hormone called testosterone, and that will involve in the production of the spermatogenesis. So if there is no testosterone, no spermatogenesis. When testosterone is liberated? Only at the time of puberty. Is that clear? Right. So FSH hormone will act on a cell called Sertolic cells, and that produces a hormone. It is an endocrine gland. It's endocrine in nature. It produces an inhibin hormone and also a protein called androgen binding protein. You know the difference between these two. Inhibin will stop excessive production of testosterone, otherwise more sperm cells will be produced. Now the androgen binding protein will maintain always the concentration. It will not go down, it will not go high. It is maintained. Is that clear? Right. So that is the one part of this introduction part of this spermatogenesis. Okay. Now we will go for what is that spermatogenesis actual processes. So now you know this difference between spermatogenesis and spermiogenesis. No? Right. So we will talk about the spermatogenesis now. Actually, inside the testis, this is the testis. The testis will have certain lobe-like structures. Like this, you will find some lobes inside. All this we will study in the next class, tomorrow's class. Okay. And inside the testis, these lobes, you will have some spirally coiled tubes will be present. Okay. And these tubes are called as seminiferous tubules. Why? Because in this tube only spermatogenesis will take place. Sperm formation takes place always in the tube called seminiferal tubules, where the tubules are inside the testes. Okay, right. So now what we do is, I am just taking this tube and doing a cross section of it. So if I cut this, I am having these kind of structures. Okay, so I am having this kind of, if it is round, I am just cutting this. I am cutting, so I am having a cross section of the seminiferal tubules here. Okay, now this seminiferal tubules here in this is a tube seminiferal. Inside you will have a lining cell like this you will have. Okay, so these cells are called germinal epithelial cells. Germinal epithelial cells. Understand? Right? This is the seminiferal tubules. I have taken a cross section. On the border, what you will have, the corner of the cell, you have the germinal epithelial cells. Okay, right. Now, the primordial germ cells. Primordial germ cells, which is the first cell for forming the sperm cell. That is called primordial. Otherwise, we can call these cells are male germ cells. Okay, male germ cells. These male germ cells will enter into this tube called, this cell will enter into the seminiferal tubules and will enter into this germinal epithelial cells. Okay, so now if this is a primordial germ cells which is diploid in nature, it is divided into two and two cells divided into four like this. Okay. One cell divided into two cells divided into four like this. Every cell has the capacity of giving rise to two cells. One cell gives rise to two. So here you got two cells. So totally you have four cells. Now this phase or this stage, it is not given in the book, but still understand, it is a multiplication process or phase. Multiplication stage. Why? Because one cell is divided into four cells. So multiplication phase. The first stage which takes place before the formation of spermatogenesis is the multiplication phase. Still now we have not studied. In spermatogenesis we have to study a cell called spermatogonia. What is spermatogonia? It is sperm producing cells. Immature sperm cells. Okay, that is what. So here I will just put process is called genesis. Cell which is involved in that process is called spermatogonia. It is a immature sperm cells. Understand these two words. What is spermatogenesis? Formation of sperm cells, immature sperm cells. What is spermatogonia? The cell which is involving in the process is called spermatogonia. Understand? Before the spermatogonia, we have primordial germ cells or sperm male germ cells. Male germ cells, which is diploid in nature, that will undergo repeated divisions like mitotic division, and it forms multiplication process and forms every cell produces. This is also diploid because by multiplication I am not told it is a reduction division. Multiplication. So everywhere you will find diploid number of cells. Okay, you got four. Among this four, 50% cell only will enter into this spermatogonia. And this 50% will support. Okay, these things are not in the book, but still you understand. Among this, only one cell, okay, will form here. So everything is happening here, inside this. All this process happening inside the germinal epithelial cells. Don't forget it. Okay, all this process I have done in the germinal epithelial cells, which is there in the seminiferal tubules. Is that clear? Now you got these four cells inside. Only one cell will grow in size. Okay, will develop. So this phase or stage is called growth phase. Growth phase. Because it is not going to divide. Growth means enlarging. It is becoming bigger. Okay, so it is not going to again further divide. So now this one cell which is going to grow, this cell is called the spermatogonia. Okay, so that one cell which is going to grow, I just put it here, which is diploid in nature. This is called the spermatogonia cells. Where can you find the spermatogonia? Okay, now this is the central part of the tube. The tube was like this. I have cut this. Now the tube is this. We are studying only on the corner. Okay, so now after all the production, mature sperm cells should come to the center. That is called the lumen of the cavity. Lumen means a tube which is inside holes. Now it should come to the central part. Okay, now from here the division starts and comes here. Now remember, very important, whenever the spermatogonia is entering inside and before it could reach the central part, it will complete its maturation process. Beginning is immature. By the time it reaches here, it will complete its maturation process. That means spermiogenesis also will be completed. Okay? Now, one more thing you remember. Whenever the spermatogonia is entering inside, it alone will not enter inside. Inside, you will have these kind of cells like this. Like this it will be. These cells are called Sertolic cells. We studied already. Sertolic cells. What is the nature of the Sertolic cells? It will produce an hormone called inhibin hormone. So it will produce a protein called androgen binding protein. What is the role of androgen binding protein? It will maintain the concentration of testosterone, as well as it will increases more, it will stop by the hormone called inhibin. Understand? Now, here this spermatogonia, it is a matured or immature cells? Spermatogonia, immature cells. Okay, it has to get mature. So, for the so if it is getting mature, it has to undergo divisions. Okay, so when it is undergoing division, what will happen is it requires some nutritions. Okay, so when it is moving from here to here, it will go among these. See, this is going among with the Sertolic cells. So suppose if this is a Sertolic cells, this spermatogonia will be fixed along with this. So to take nutrition, till it is going to the central part, it needs the help of the Sertolic cells. Understand? When the spermatogonia, it start dividing here and moves into the central part, it will become mature. For that it needs some nutrition. So it will go enter along with the Sertolic cells. So the Sertolic cells will, this is a Sertolic cell, which will supply the nutrition for this spermatogonia. All the nutrition supply. So till maturation, it needs the help of the Sertolic cells. So Sertolic cells not only gives an hormone called inhibitin, not only gives a protein called androgen binding protein, it also nourishes the developing sperm cells. It gives nutrition, it feeds the sperm cells. Okay, that is the reason these spermatogonia cells will go along with the Sertolic cells to the inner cavity, that is to the center of the speminiferal tubules. Is that clear? Right. So now this spermatogonia, now the second stage is the spermatogonia will divide by a process called mitotic process. Okay, the spermatogonia cells will divide. Now here we are starting from here to here. Okay, we studied what happened inside the germinal epithelial cells. What happened? The primordial germ cells, which is converted or undergoing multiplication division to form 4 equal number of the spermatogonia cells. Among them, 50% of the cell will undergo growth phase and that will form spermatogonia. So from where the spermatogonia has come? From which cell spermatogonia has come? or which cell gives rise to spermatogonia? Primordial germ cells, where it is developed in the seminiferal tubules of the germinal epithelial cells. Now it will move to the central part of the seminiferal tubules among with the sertolic cells. You know the reason why it is, because it supplies nutrition, so it takes the nutrition for the development. Is that clear? Right. So in this process is this spermatogenesis, where immature sperm cells are produced. In that, the spermatogonia, which is diploid, will undergo mitotic division to form the equal number of diploid primary spermatocytes. Okay, this is everything happens after puberty. That is what the important is. That means this process has done before puberty. Spermatogenesis alone will take place after puberty. You got the answer for now. What happened before? That means primordial germ cell start to divide, making this spermatogonia. Ultimately, this cell will be giving a cell called spermatogonia. So this cell is produced by the primordial germ cells. That means primordial germ cells start to divide at the first week of gestation. First week of gestation itself, when a male is formed in the womb of the mother, this primordial germ cells start divide and gives you spermatogonia. And all the spermatogonias are now, it is in the germinal epithelial cells. Then a male is born with spermatogonia. That is what the beginning itself I told. Okay, spermatogonia. Female, primary oocyte. Because egg mother cell was there, not spermatogonia, egg mother cell. So that egg mother cell is converted into primary oocyte. Everything happened before birth. After birth, you have spermatogonia. Till puberty, nothing is done. No layers, nothing is formed. It is just arrested. After puberty, only all the process is done without any break. We studied there, that was discontinuous process. Here it is continuous process. Why? Because from puberty only it starts. No fertility, nothing. It is not doing anything in between. So starts dividing into primary spermatogonia. Understand, right? So now again this, I told you 50% only will birth. Another 50% will support it. So this primary spermatocyte again will undergo a reduction division called first meiotic division, and this forms another, that means you take this 4 so that you can understand. So you got 4 haploid, because reduction division, haploid secondary spermatocytes. Is that clear? Secondary spermatocytes. Okay. Now again it will undergo division, and that means here you got the secondary spermatocyte, and this forms the structure. One more here, that is the again haploid. So this is what again further division it will lead to spermatid. Spermatid. Now up to this the divisions are common, equal. So everywhere we studied there, it is an unequal division, where in female, but male it is always equal division. See, spermatogonia divides into 2 diploid primary spermatocytes. Then first meiotic division, this primary spermatocyte will again convert it into secondary spermatocyte. Okay, right, which was diploid, now become haploid. Now all this will form into the spermatids. Clear? Right. Now from here the process starts is spermiogenesis. Spermiogenesis. Whenever the spermatids are formed, okay, then it will undergo a process called maturation process called spermiogenesis. Then here this 4, I will draw here, this 4 things which is haploid will be producing. All these are haploid, all these are called spermatid. Okay, will undergo a structure called, it forms an head, it forms a neck, then it forms a tail. So like this, the sperm cells are formed. So these are called the spermatozoa. The difference between spermatic and spermatozoa is all are haploid only, but the difference is it is round, circular. But here the spermatozoa are having the structures for the movement. That means it is differentiated into a neck and the tail part. Is that clear? Now after this class we will be studying detail about the structure of the sperm cells. How the sperm cell will look like. This one. How it is. Understand? So this much you are very clear about this. Spermatogonia undergo mitotic division to form primary spermatocytes and meiotic one form secondary spermatocyte, and then it forms into spermatids. Okay, now the spermatids will be converted into spermatozoa. Clear? We'll have a quick recap on what we studied so far in spermatogenesis. Okay, diagram part of it. Now already we have studied about the primordial germ cells. What we are studying is before the birth of the individual. In the testis, the primordial germ cells, actually every process is taking place inside the tube called seminiferal tubules, which is lined with germinal epithelial cells. So inside this germinal epithelial cells, this primordial germ cells will undergo multiple division to form 2 and 2 cells form 4. So all these cells are called the spermatogonia cells. Okay, now among this 50% only will involve in this process. I already told you. Why 50 and another 50 will be supporting it. Okay, so now here we got this only one cell which among the spermatogonia. Now one cell here, it is going to become differentiated. That means it is going to get mature and it is developing under the process, and after maturation it will enter into the central part of the seminiferal tubules. Okay, so after maturation, so that means till it reaches the central part, it requires the help of the Sertolic cells. Okay, without Sertolic cells it cannot move inside because it will take up the nutritions. Now once it is getting mature, that means once it becomes spermatozoa, it leaves the Sertolic cells and it is liberated into these areas. That is the central part of the seminiferal tubules. Okay, that means if you talk about this tube, you find the sperm production in different stages. Here you will have spermatogonia, then you will have primary spermatocyte, then you will have secondary spermatocyte, then you will have spermatid, then you will have spermatozoa. So in each tubes of the seminiferal tubule, you will have different stages of the sperm production. Understand? Everywhere you will find different stages. So ultimately when the sperm cells are getting matured and liberated into the central part of the seminiferal tubule, it will leave the Sertolic cells and come out. So that is called semi-insemination or seminations, right? Now here we find a structure or a word, an important word in the book called spermiation. Spermiation. So nothing, spermiation means is a 2-mark question also. We can find afterwards. Spermiation means the sperm cells mature and they are liberated into the central cavity of the seminiferal tubule. That is called spermiation. Okay, sperms are liberated. But from where the sperms are liberated? From the Sertolic cells. Understand, that word is not there in the book. But it's still you have to understand. So in spermiation, after sperm is mature, it is liberated into the central part. Called spermiation. Sperms are liberated. Sperms are active now inside the tube. They are ready to fuse. Now, how from where the sperm has fallen? It is fallen from the Sertolic cells. So when the Sertolic cell leaves out, till that it holds the hand. For till maturation, it is holding the hand, supplying all the nutrition. Once it gets matured, it just leaves it and comes to the central part of the seminiferal tubules. Understand, right? So now, this one spermatogonia which is diploid will undergo mitotic division to form, mitotic means equal division, to form diploid primary spermatocytes. Okay, this diagram what is there in the book, same you can draw for the exam also. What I explained is for your understanding purpose. Okay, right. So here you got one spermatogonia which is going to mature. Okay, why? Because half of the thing is going to mature. Because we have here 4. All the 4 not possible. 50% is possible. So now it becomes first meiosis division. In first meiotic division, reduction division takes place. So you get 2 haploid, equal number of secondary spermatocyte. When we studied about the oogenesis, what we studied? One is large, one is small. Okay, but here it is equal. So it is called equal division. Next one. Again, each now there only one secondary oocyte has gone for another development. But here both the secondary spermatocyte will undergo second meiotic division, and each one will give 2 haploid spermatids. So every secondary spermatocyte will give has the capacity of giving 2 haploid spermatids. So if it is 2, it is having 4 haploid. Okay, spermatid. Now the spermatid will undergo spermiogenesis, maturation process to form spermatozoa. Then it is liberated into the central part called spermiation. This much is clear? Understood? Right. So very important one. In your book here and there the content will be, but I have given you in a sequence. So please understand and read it once. Clear? Right. Now we will talk about the structure of the sperm cells, and with that we will complete it. Okay. With that we will have your recap also along. So now we got. Now tell me the matured stage. What is the immature sperm cell called as? Spermatogonia. Very good. What is the matured sperm cell is called as? Spermatozoa. Very good. What process is involving in maturation process? Spermogenesis. What is the immature process? Spermatogenesis. Understand? So there is a spermatogenesis. Here it is spermiogenesis. Okay, next one. As we studied about the structure of ovum, we will study about the structure of sperm cells. Suppose if they are asking spermatogenesis separately for a public exam or oogenesis separately, if they are asking to write the structure of sperm, then you write. Otherwise, write only the spermatogenesis alone. Same for oogenesis also. Okay, right. Before the structure of sperm cells, I would like to ask you one question. What is gametogenesis?
Formation of gametes.
Gametogenesis. Formation of gametes. Because genesis means formation, I told you already. In male, what is produced? Sperm cells are produced. So sperm is the gamete. In female, it is the egg or the ovum is produced. So in gametogenesis, the process of a formation of sperm cells and egg cells takes place. Okay, right. If sperm has to produce, okay, or egg cell has to produce, all these are matured one. Sperm is a matured, spermatozoa, egg, ovum is a matured one. Correct, no? So if that has to produce, what cell division? Whether it is mitosis or meiosis? No, any one. Either it is mitosis or meiosis. I am talking about matured process. Whether it is meiosis or mitosis? Meiosis. So in gametogenesis, that cell division takes place. Which is very important is meiosis. That's all 2 mark question and 1 mark question. Define gametogenesis means you have to write this and one. The cell division called meiosis. Why meiosis? Because matured process. Though it is happening mitosis, meiosis, only mitosis gamete will not produce. Mitosis is called only equal division. Okay. Meiosis only will produce sperm cells and egg cells. So in gametogenesis, meiosis play a key role, vital role in producing sperm cells and egg cells. It was given beginning of this content. Okay. I want you to answer. That is why I do not teach that paragraph. Now we understood what is gametogenesis, why meiosis. Without meiosis, no sperm cells. Without meiosis, no egg cells. Is that clear? Right. When you talk about the structure of sperm cells, actually the structure of sperm cells, the sperm cells are microscopic in nature. What do you mean by microscopic? You can see only through the microscope. Okay, that is what microscopic in nature. And they are flagellated. Flagellated means it is having a tail-like structure. Flagellated, or it is having a tail-like structures. It is having a tail-like structure. For flagella that we will talk about. Okay, right. Now the sperm cells are motile cells. What is motile?
Movement.
The sperm cell can move. What about egg cell? Egg cells are non-motile. But how it is moving in the fallopian tube? It is liberated in the ovary. It has to go to the tube and come to the uterus. How it is moving? By a movement called ciliary movement. Sperm cells are. If the sperm cells are not there in the fertilization, menstrual cycle is taking place. Then how the egg cells are moving? By a kind of, see, if it is a tube, the tube will have hair-like growth. Okay, once when egg falls on this, the hair will move. So ciliary movement. That movement is called ciliary movement. When a microorganism, bacteria, enters into the nostril, how it is going into your tract, respiratory tract? Because of this movement called ciliary movement. Okay, so this is hair-like growth which will move like this, so that the egg will be moving. Is that clear? So that is what the movement. So it is called motile cell, but still it moves with the help of the cilia. Okay, right. So now we will talk about the structure of the sperm cells. Now the sperm cell structure will have three parts. This one is the egg part. Okay, the egg will have a small. Egg is having two types, two things in this. A small cap-like structure you will find here. This one is called the agrosome. Listen, agrosome. Okay, the first part in the egg is called agrosome. And these agrosomes are formed from the Golgi bodies. One mark question. Agrosomes are formed from the cells will have Golgi body. From those Golgi bodies, agrosomes are formed. And this agrosome will have an enzyme called proteolytic enzyme, or otherwise called as sperm lysine. It is helping to destroy the protein, or the sperm will lyse something. Okay, it's called sperm lysine. Lysine means to destroy, or it is otherwise called as hyaluronidase enzyme. So this agrosome will produce from Golgi body as an enzyme called hyaluronidase enzyme, which is also called as proteolytic, or otherwise called as sperm lysine. Don't forget it. Sperm is not going to lyse. Sperm is making something to lyse. And this agrosome is very important for entering into this egg cell for fertilization process. Is that clear?
Yes, sir.
About agrosome? Right. Second part, it is having the nucleus. The nucleus is oval shaped or flat. That's all about the nucleus. Is that clear? So two parts, agrosome and the nucleus. You know about the agrosome. Okay, so this agrosome is very much important for entering into this egg cell. Is that clear? Because when it's entering into the egg cell, we studied the zona pellucida and carno radiata fixed by an enzyme, an acid called hyaluronic acid. So this will be lysing this and enter into this. Is that clear? So that is what the fertilization agrosome, which is always involving in the fertilization. Second one, it is having a very short neck that you cannot see at all. You cannot differentiate it. So that is called the very short neck. They will have which you cannot see properly. So that is what the neck is. Understand, right? So now this short neck will have, I just draw like this for you to understand, but do not have, that neck cannot be visualized. Okay. Here you will find a centrioles like this. Centrioles are very much important to identify. If the centriole is located, you can easily identify the front part will be nucleus. To identify the nucleus, centrioles are important. By seeing the centriole, you can differentiate or determine where the nucleus is located in the cell. Okay, so this is a centriole which is also involving in cell cycle. You studied last year botany, 11th standard, cell cycle. So the centrioles are formed, all the chromatin fibers are formed here, spindle fibers formed, all this. So the centrioles are very much important. Now there are two centrioles will be there inside the neck, but neck is not this much big. For your understanding, I am telling about the centrioles. Okay, when you draw the diagram, don't show the neck there. Right. So now it is having the centriole. The centriole which is close or forward is called the proximal centrioles. Okay, the centriole which is forward, close by to the nucleus, are called proximal. And always this is involving the centriole which is towards the nucleus will involve in the cell division to form zygote. Okay, one more question. Which one is undergoing cell division to form zygote? The centriole which is close, proximal end. Is that clear? Then the centriole which is back, second one, that is called distal end or distal centrioles. Distal is back side. This forms a region like this. This is called axial filament. Axial filament. Understand?
Yes, sir.
The centriole which is forward goes for division, forms zygote. The centriole which is back forms a distal end, that is forming an axial filament, central filament. Now it is having the first part is the head part. Second part is the neck part. That is what we study. Okay? And it is having the middle piece. This piece called middle piece. Now in this middle piece, you will find this structure like this. So that structure is called mitochondria. Okay, where the mitochondria is now, it is encircling the axial filament. So it is called mitochondrial spindle, or otherwise called as Nebenkerl. Okay, this understand. Okay, right. This is an important one. And next one, this mitochondria will always supply ATP, energy, for the movement of the sperm cells. Okay, then the third part of this, it is having a tail-like structure. This is the tail-like structures for the movement. Okay, and this tail-like structure is formed from this axial filament. Axial filament only it is formed. So it is called axonema. Axonema. This is what the structure of sperm cells. Around 200 to 300 millions of sperm cells are produced per day. Like this, in testis, we produce 200 to 300 millions of sperm cells in after puberty, that is from 10 to 16 years onwards. It is continuous till your death. So male is always fertile. They remain fertile throughout the life. Female only for 48 to 50 up to menopause, but male remain fertile for long. Understand? So that is what the structure of the sperm cells. And overall, the spermatogenesis completion takes place. Spermatogenesis, formation of sperm cells, takes place actually 74 days, but in your book it is given only 64 days. From the top, germinal epithelial cells to the center, it will take around 64 days to complete it, or 74 days to complete it. Understand, that means one spermatogonial cell undergo differentiation to form spermatozoa. It takes place 74 days actually, but I don't know why it is given 64 in your book. It is given as 64, but if you refer all the books, you will have 74 to 64. Okay, so maximum it is given, so you remember only 64 for the time. Understand? Any doubts? Right. Take your note. You will write your recap now. Question number 1. What you study the same you will write it. Question number 1. Which cell undergoes first meiotic division in spermatogenesis? Which cell undergoes first meiotic division? Which cell undergoes first meiotic division? Which cell undergoes first meiotic division? Number 2. Which cell comes earliest in the sequence of sperm production? Earliest in the sequence of sperm production. Which cell comes earliest in the sequence of sperm production inside the seminiferal tubules? Third one. Which cell have haploid number of chromosomes? Which cell has haploid number of chromosomes? I will give you an option. Primary spermatozoite, secondary spermatozoite, spermatid, B and C. Primary spermatozoite, secondary spermatozoite, spermatid, B and C. Which are the cells will have haploid? Next one. During spermatogenesis, which stage is the first contain haploid number of chromosomes? In spermatogenesis, which stage of the cell contain haploid number of chromosomes? Haploid. Very simple. Last question. Spermiogenesis is. What is spermiogenesis? Spermiogenesis. Water is transformed into what? That is what? In spermiogenesis, what is transformed into what? What is transformed into what? Check the answer. Meiosis 1 division takes place in primary spermatocyte. Number 2. The earliest sequence of the sperm production, it is the spermatogonia. Which are the cells will have haploid number of chromosomes? That is secondary spermatocyte and spermatid, and even spermatozoa also correct. Now, which stage the first contain haploid number of chromosome? Secondary spermatocyte only will contain haploid number of chromosome. Before that only diploid. Correct? In spermiogenesis, what is transformed into what? Spermatids are transformed into spermatozoa. Clear? Right. So next class we will be studying about the male and female organs in detail.